Hello. We're continuing with my life science in a nutshell. Notes and say hi, Ruben.
Well,
We were learning about the components of the cells, um, interior. And it's cytoskeleton. So, um, there are proteins which are called septin and spectrin. So we're learning about those. They associate with the cytoskeleton, namely the three parts of the cytoskeleton, which are microfilaments, microtubules, and as well as intermediate filaments. So we learned about the what each one is. So these proteins, which associate, help with the functioning of these filaments in the cytoskeleton. So this is a picture of septins. The green is the stainage of the proteins. So this is what it looks like right here. It has a GGP binding domain, so it uses energy to work, and it also associates with other proteins as well. And it has a huge role in uh, cell replication. Ruben, you want to read this? The predominant.
the predominant functional conformation is characterized by a low turnover rate, frozen state during which the septins are phosphorylated. Structural changes require a destabilization of the septin cortex fluid state induced by dephosphorylation,
Dephosphorylation. Dephosphorylation.
dephosphorylation prior to bud emergence, ring splitting, and cell separation. Functions: the septins act as a scaffold recruiting many proteins. These protein complexes are involved in cytokinesis,
Cytokinesis.
cytokinesis, chitin, the deposition, cell polarity, spore formation, and morphogenesis, checkpoint, spindle alignment, checkpoint, and bud site deposition, cell.
Selection.
So,
Okay. So basically, it's a very important protein in the replication of cells and spindle alignment, morphogenesis, or the changing of the shape of the cell, and where the cell is gonna bud or split, and cell polarity, meaning the different shape of the cell, the ends and such, and pulling of the spindle. When you have replication and you need to pull the two different daughter cells apart in separation, and cytokinesis, splitting of the cell's interior and the cytoplasm and its organelles, etc., as well as the nuclear material. So cytokinesis, in yeast, when you have budding, it's driven by two septins, in the process of, and it recruits and uses actomycin as well in a ring formation to help with that, with the contracting. And you have vesicle fusion, etc., with the plasma membrane. So, so it interacts with the membrane of the cell as well. In contrast to septins in yeast, in contrast with other cytoskeletal components of metazoa, septins do not form a continuous network in metazoan cells, but several dispersed ones in the cortical cytoplasm. So earlier we saw the cortex, um, which is the referring to the nucleus and what it contains. So we learned that the cell cortex, or the actin or actomycin cortex, is in the inner layer of inside of the plasma membrane of the cell periphery. It controls the cell's membrane's behavior and its surface properties. So the spectrin is found in this cell cortex, as well as the actin and actomycin. So, excuse me, earlier I got confused. So this is the cell cortex, so it's inside the cell, not necessarily the nucleus. That was nuclear lumina. That was inside referring to the inside of the nucleus, lamina, which is shown right here, inside, and that's made up of mostly intermediate filaments, which are the third part of the cytoskeleton, nuclear lamina, as we learned right here in this chart. Okay, so the spectrin again are integrated with the actin bundles as well as microtubules. The septin are localized as well in mitochondria, called mitochondrial septin. Micrographs of purified filaments raise possibility that septins are organized in parallel to the bud axis. So it's 10 nanometer striations seen on electron micrographs, showing lateral interaction between the filaments. Okay, so the next kind of protein we're learning about is called spectrin. Ruben, go ahead.
Spectrin is a cytoskeletal protein that lines the intracellular side of the plasma membrane in eukaryotic cells. Spectrin forms pentagonal and hexagonal arrangements, forming a scaffolding and playing an important role in maintenance of plasma membrane integrity and cytoskeletal structure.
Cytoskeletal.
Alpha 2 spectrum in green under the plasma membrane of rat neurons.
In tissue culture, shown here in this picture, which is shown with a confocal microscope and immunofluorescence. It looks very beautiful. The nuclei of the cell is revealed in blue, which is the DNA, stained with a dye. And I believe the green is with the spectrins. Okay. And then here we have a diagram. It says beta spectrin, alpha spectrin, actin. It's just showing glycoprotein, tropomyosin, spectrin is shown here. We're continuing in the next video. Thank you guys. Bye.
Bye.